Okay, so we just went through the steps of glycolysis. We talked about each of the 10 steps. We talked a little bit about the logic of those steps and the results in terms of the um, out output of glycolysis. Now all we're going to do is go through each of those steps. I'll point out some details. Again, I'm not going to go through all the details. I'll pull out some things that refer back to principles that we've learned before in terms of catalytic mechanisms, in terms of um, some of the good chemistry, redox reactions, and things like that. But we aren't going to go into a detailed explanation of each of the mechanisms of the 10 steps in glycolysis. This is something that is very fairly well characterized, and so we could do this for each of them. And again, if you took Dr. Young's biophysics, that might be something that you would do. And in looking at the energetics here, I'll simply point out this is looking at the delta G for each of the 10 steps in glycolysis. And note that the ones in which we invest ATP have a large negative delta G. That kind of makes sense. We're using the energy from the hydrolysis of ATP in order to drive these reactions, so the net result should be negative. But also note that there are, and well, let me just say here, remember, third step, this is the last time we invest ATP. But yet, after that step, there are several reactions that are endergonic, all right? And yet, by manipulating the concentration of products and substrates, you can change the actual delta G here. Oops, no, that arrow should be in the other way. So here, for instance, the aldolase reaction, the standard delta G is positive 23.8, but by manipulating the concentration of the products, specifically by converting dihydroxyacetone to glyceraldehyde 3-phosphate, and by immediately pushing glyceraldehyde 3-phosphate into the dehydrogenase step, you decrease the concentration of the products, right? And if we remember from Le Chatelier's principle, we therefore drive the reaction in the forward direction, and we can make the actual delta G, in this case, negative 0.4 kilojoules per mole, right? So in this case, by manipulating the concentration of products, we make a um, endergonic reaction, exergonic, or essentially reversible in this case. And we'll see this a couple more times, in this step, um, here, uh, in which we're taking what would be endergonic reactions and making them either reversible or exergonic. So one of the tricks is to manipulate the concentration of products, mainly by keeping the steady state concentration of those products relatively low so that you can change the actual delta G of a um, of a reaction. Okay, so first step of glycolysis is the hexokinase reaction. We're taking a glucose and we're simply adding a phosphate to carbon number 6 there that uses ATP as a coenzyme in order to make that reaction work. The second step is to take this glucose 6 phosphate molecule and isomerize it to a fructose. And remember, let's see, this has this is carbon number 6. So 1, 2, 3, 4, 5, 6 carbons here. And then here, 1, 2, 3, 4, 5, 6 carbons here. So we aren't changing the number of carbons, we are only changing the ring structure, right? By moving the carbonyl group to from an aldehyde to a ketone, right? And I think, good, I do have the mechanism here. So just looking at this mechanism, here we have this glucose 6 phosphate. We're going to open that up. And then what's going to happen? This is the enzyme over here, phosphohexose isomerase. There is this compound here that is going to remove a hydrogen from the substrate. Now, we know that that is then an example of what? Oh, wait. Right, general base catalysis, right? And so you have the enzyme, it's not water, so it's something other than water that is extracting a proton from the substrate. So this is general acid base catalysis, right? And so here we have that hydrogen that's been extracted, that's going to form this cis and diol intermediate. Note the hydroxyl, carbon-carbon double bond, another hydroxyl, well-known intermediate. This is basically a way to just shift that double bond. And so you see the carbonyl, the carbon-oxygen double bond is here. You move to this carbon-carbon double bond, and then this is going to collapse. Where are we? Here we are. This is going to collapse down to form a carbonyl double bond, and then um, you restore the enzyme. And so in this process, what have we done? We've basically just shifted the double bond from the carbonyl group on carbon number 6 to the 2 carbons and then ultimately over here. So a very common way to move a double bond um, from one carbon to another. We'll see that cis and diol intermediate, and I'm sure you guys do that in organic chemistry quite a bit. If you want to, go ahead and impress Dr. Young by pulling that out as a mechanism of action. Next step is to um, phosphorylate carbon number 1 here. The enzyme that does that, phosphorate fructose kinase, we kind of went over that already. We're going to invest a molecule of ATP. We talked about the regulation there. In order to do that, you're going to form 1,6 fructose 1,6 bisphosphate, right? So yeah, phosphate on carbon number 1, a phosphate on carbon number 6, and it's a fructose molecule, so there you have it. And this is the rate limiting step of the entire pathway, and we talked about that already. The next step is kind of cool. This is the Adolase reaction in which we actually split this compound. Here we take fructose 1,6 bisphosphate, and we're going to chop it up into two 3-carbon structures, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Note here that 3 carbons on each, right? These are isomers of each other. The only difference here is really the position of the carbonyl group. Position of phosphate as well, but details. And so here, this is obviously an aldehyde, which means that the carbonyl group is at the end. And this one is a ketone, which means that the carbonyl group, well, since it's 3 carbons, it has to be in the middle here, right? And so this is going to break this up into two 3-carbon structures. This is a pretty kind of neat mechanism. Remember, we're operating according to Occam's razor. And so the question is, why do we need to do the isomerization reaction in step number 2? We don't, if we're just looking at things in terms of delta G, the delta G of the reaction, glucose and fructose is roughly the same. But if we understand the mechanism of how aldol condensations work, then we can understand why the products would result in something that's easier to work with if we split glucose versus, split fructose versus splitting glucose. And that's shown here. Um, Remember, aldolase does what is essentially an aldol condensation reaction in reverse. I'm sure you've learned about aldol condensations in organic chemistry, or you're learning about it now. Here you have a carbanion that acts as a nucleophile, and it is going to attack the electrophilic carbon of a carbonyl group, and that forms a new carbon-carbon single single bond. I will note that the bond formation is what? It's at the carbon that is adjacent to a carbonyl carbon, right? And so if you look at this carbonyl carbon, you go one carbon over, that is the bond that's going to be formed. Now, if we do this in reverse, then the bond that's going to be split is that same one, the carbon that is adjacent to the carbonyl carbon. So if we look at glucose, look at this. Here's the carbonyl carbon. If we're doing an aldol condensation, then the bond that will be broken in glucose is the carbon that's adjacent to the carbonyl carbon, which would be right here. So in glucose, we would do the cleavage right at carbon number 2. Now in fructose, what happens? Okay, well, here's the carbonyl. The carbon adjacent to it is right here, and so the cleavage will be right there, right? So in glucose, the product of the aldolase reaction would be a 4-carbon structure and a 2-carbon structure. Whereas in fructose, you get 2 3-carbon structures, which are essentially isomers of each other. And so by doing an isomerization reaction, which is energetically neutral, uh, earlier in the process, then we can actually save 6 steps later in the process by forming 2 3-carbon structures instead of a 4-carbon structure and a 2-carbon structure, right? So this allows you, by making an initial investment in an enzyme, not even really delta G, it allows you to simplify things in the entire payoff phase where you can use the same set of machinery, the same set of enzymes in order to process these compounds. So really neat in the way the chemistry and the biology kind of come together and to form something that works and makes sense. All right, so here's the aldolase reaction. Again, there's 2 3-carbon structures that are isomers of each other. Rather than having to process 2 individual compounds, we can simply isomerize one to the other and process that. And so that's what this is the aldolase reaction. So that's what this triose phosphate isomerase does. It takes dihydroxyacetone phosphate, converts it to glyceraldehyde 3-phosphate. All we're doing is moving a carbonyl double bond, and so we can see again, we're going to use this cis and diol intermediate in order to make that happen. And so from this, we get 2 glyceraldehyde 3-phosphates that can be processed in the payoff phase of the glycolytic pathway. And so, first step is the dehydrogenation of glyceraldehyde 3-phosphate. So here, dehydrogenation is going to pull off electrons, right? And so we are going to do that by using, in this case, a molecule of inorganic phosphate. So most of the phosphate that we've seen has come from ATP. This one, again, since we aren't investing ATP anymore, it doesn't come from ATP, but it is instead coming from an inorganic phosphate that's floating around in the cytosol. And so remember that these electrons, the high energy electrons, are represented by these carbon um, um, hydrogen bonds, these hydrocarbon bonds. And so we see that we're going to take the electrons from this particular hydrocarbon bond, we're going to transfer them to NADH and then replace it with a phosphate group. The next step is to pull off those phosphates. Oh, and this forms the 2 NADH that represent a good portion of the energy that is pulled out of this process. The next step is substrate level phosphorylation. So this is phosphorylation that occurs at the level of the, an enzyme. An enzyme is literally pulling the phosphate off of something and putting it onto ATP. We'll see that that compares to um, uh, oxidative phosphorylation, which is a process by which you're using the energy of a concentration gradient in order to drive ATP production from ADP, an inorganic phosphate. And so we have this 1,3-bisphosphoglycerate. We're going to pull the phosphate off, add it to ATP, and leave 3-phosphoglycerate in the process. So this is the first time that we produce ATP um, in this process. The next step is to move this phosphate to carbon number 2. Again, this is a mutase reaction, which is essentially a isomerization reaction. So it's cheap energetically, and this is something that has a payoff 2 steps down the line. So it doesn't pay off immediately, but it makes sense in the last step of the of the process. Um, the next step is a dehydration reaction in which you're going to remove a water from this compound and form phosphoenolpyruvate. And this PEP has a lot of energy associated with it. And we're going to remove this phosphate in order to generate the last 2 molecules of ATP. Pyruvate kinase is the last one. This is again substrate level phosphorylation. You form your ATP out of here, and we're going to take the phosphate from phosphoenolpyruvate, remove it. You're going to be left with pyruvate and the 2 molecules of ATP that are formed from the 2-glyceraldehyde 3-phosphate that we started with. And this is another example where by putting this phosphate in carbon number 2, what happens is that you convert the enol form of pyruvate. It will spontaneously tautomerize into the keto form. And I think you've done tautomerization in organic. Note the kind of directionality here. This is something that happens quickly and spontaneously um, upon the production of this pyruvate. As a result, what have you done? You've decreased the product of the pyruvate kinase reaction, which is the enol form of pyruvate. And so you do what? You drive the reaction in the forward direction and you pull out a lot more energy from that reaction than you would normally have. And so then again, by doing uh, a isomerization reaction earlier in the cycle, you have a payoff that pulls more energy out of the system at the last step in the cycle. And, at the end of that, you have this pyruvate in the keto form. This actually has a lot of energy associated with it. That energy is found in the hydrocarbon bonds that are over here, and you have your uh, net 2 molecules of ATP and NADH that is formed. As I said before, this is regulated, and it's regulated at the areas of dramatic changes in the delta G. So hexokinase, phosphofructose kinase, pyruvate kinase. We don't talk about this. We would get to it in an advanced biochemistry class, but the areas so gluconeogenesis is the biosynthetic pathway. So this is taking pyruvate and going back to glucose, and it utilizes a lot of the same enzymes as in reactions as glycolysis, except for those areas where there's a dramatic change in delta G. And so when you have a shift in delta G, the cell has to come up with some other way to bypass it. And so in the pyruvate kinase reaction, there are actually 2 steps: pyruvate carboxykinase and pyruvate carboxylase that happens. In the phosphofructose kinase step, which is the last time we invest ATP, that in gluconeogenesis is fructose 1,6-bisphosphatase. And then in the hexokinase step, that last path is glucose phosphatase. And so in the places where there are um, uh, delta Gs that are reversible or close to reversible, you can use the same pathways, but when there are dramatic shifts in delta Gs, it's irreversible reactions. So you have to find something that bypasses it. And I will say we'll talk a little bit about this later on. It's a little bit complex, but the um, the factors that regulate this pathway, particularly at the level of phosphofructose kinase, it will be inhibited by high concentrations of ATP, and it'll be activated by high concentrations of ADP. Why? Well, if you have high concentrations of ATP, that indicates that your cell isn't using the energy that it has. And so it doesn't need to create more energy, so you'll downregulate the process. If you have high concentrations of ADP, that indicates that the cell is actively using ATP and then it needs to make more. And so that will positively regulate those enzymes. And so we'll see this throughout that ATP, citrate, NADH, they tend to downregulate the cellular respiration pathway, whereas ADP, NADH, and other kind of metabolites tend to upregulate this pathway. And we've talked about glucose and how goes through the glycolytic pathway, but there are other entrees into this pathway. So if you have something like sucrose, or if you're just consuming table sugar, sucrose is a disaccharide that consists of one fructose molecule and one glucose molecule. So disaccharide gets split, glucose goes into the glycolytic pathway, and then the fructose can directly be phosphorylated um, uh, to form fructose 6 phosphate, and then you go through the pathway, right? Lactose, which is a glucose and a galactose, gets split. Glucose goes in, the galactose gets isomerized to glucose 1 phosphate, and then it gets mutated to glucose 6 phosphate so it can feed into the pathway. Glycerol, is that in here? Yeah, so if we take a triglyceride, the glycerol gets cut from the triglyceride. We'll deal with the fatty acids later. They go directly into acetyl-CoA formation. But that glycerol is basically a 3-carbon structure. It kind of looks like glyceraldehyde 3-phosphate. So it gets converted to glycerol 3-phosphate and then goes into dihydroxy acetone phosphate, which then gets isomerized to glycerol aldehyde 3-phosphate. In the process, you actually generate an FADH2, which has, bucket loads of energy. So you actually get a little more energy out of that process. And so there are multiple entrees into this. It's not just glucose. There are multiple ways in order to um, to get there. Okay. The last thing to talk about, this is running long, um, is what happens after we form this pyruvate. And there are two possibilities. If there's oxygen present, then you tend to go into the citric acid cycle and oxidative phosphorylation in the mitochondria. Now, from that, you are able to extract another 30 odd ATP out. And so you can get 32 ATP from oxidative phosphorylation and cellular reproduction um, if oxygen is present. Without oxygen, you have to make some compromises. In animals, we make lactate, and in yeast, we make ethanol. Now here, you still only result in 2 ATP, and you actually have to invest a little bit in order to convert these things to lactate or ethanol, and we'll see that. So you're going to put in a little bit more energy in doing that. Okay, so that's shown here. Um, in anaerobic metabolism, you have glucose, you go to pyruvate. Here, the pyruvate in animals can go to lactate, which is a 3-carbon structure. Sure. Okay. You don't want to see that. Um, and then in yeast, you're going to go fermentation, ethanol. This is a 2-carbon structure. So you generate a molecule of carbon dioxide in that process, right? So those are the bubbles that you get from the carbonation process when you're making like ginger ale or bread or something like that. That is the yeast taking this pyruvate in anaerobic conditions, making ethanol out of it. That ethanol is what we consume, particularly when you're under quarantine with 2 kids. Uh, Consume a lot of it, and the carbon dioxide that's produced. The role of both of these in terms of anaerobic metabolism is kind of interesting. It is to regenerate NAD plus from NADH, right? Now remember, oh, that's said here. So this is to make NADH. So remember, the way in which you make NADH is to take NAD plus and dehydrogenate it to form NADH, right? Now, what's key is that anytime you make a molecule of NADH, you lose a molecule of NAD plus. So at some point, you're going to get to a place where your NADH ratio um, is much higher than NAD plus. When that happens, then the energy that you get to make that NADH won't be sufficient in order to find the NAD plus. And so the NADH over NAD plus ratio gets very high, much, much greater than 1. Now, this is a dangerous thing, right? And so the cell has to maintain that ratio into something that's acceptable. And so they regenerate NAD plus from NADH. And so here, if you see, this process is, so here we are making NADH from NAD plus, and then in the absence of um, oxygen, then this isn't a good one. In the absence of oxygen, what we are doing is taking that NADH and regenerating NAD plus, right? And so here we're taking pyruvate. This is in lactose formation. We're taking pyruvate, and then which is and oxidized, and we are reducing it to lactate, right? Now, this is a little bit counterintuitive, right? Because here we went through a whole lot to reduce or to oxidize glucose to pyruvate, right? We went through 10 steps of the glycolytic pathway. And yet, here we are, after we've oxidized it, we are going back and then reducing it. And remember, you can count the carbon hydrogen ratio to determine the reduction level. So clearly, this has 5 hydrogen to 3 carbon, and this is 1 to 1, right? And so all that time that we spend oxidizing this pyruvate, we just go right back to reduce it, but that allows for the regeneration of NAD plus, that allows for the glycolytic cycle to continue. Okay. Whew. So that's it for glycolysis. The result here is that we've gotten a little bit of ATP. We have NADH that will feed into the electron transport chain. And we have 2 molecules of pyruvate that we're going to process in the Krebs cycle or the citric acid cycle. And so the next chapter is going to look at what happens to that pyruvate and how we process it down the line. Okay, this is longer than I like, but a lot of good information.